Good morning, all of you. I am Mahendra Saini, Assistant Professor, Department of Pharmacy, Suresh Gyan Vihar University. Today I will discuss about the topic is AIDS, acquired immunodeficiency syndrome. First of all, discuss the learning. First of all, discuss the learning objectives of this topic. First of all, being on the content in this chapter, the reader should be able to like introduction of acquired immunodeficiency syndrome, etiology of AIDS, pathogenesis of AIDS, sign and symptoms of AIDS, mode of transmission of AIDS, diagnostic test and diagnostic test of AIDS, and last one learning objective is treatment of AIDS. These are the learning objectives of this topic. Next one, learning outcomes. First of all, by the end of the lecture, the student should be able to understand like introduction of AIDS, and introduction of AIDS means definition of AIDS and etiology of AIDS. Next one, pathogenesis of AIDS and sign and symptoms of AIDS and mode of transmission of AIDS and diagnostic test of AIDS. And last one, learning outcome is treatment of AIDS. These are the learning outcomes for this topic. First of all, introduction of AIDS. First of all, introduction of AIDS. Acquired immunodeficiency disease. AIDS means acquired immunodeficiency disease, and it is a disease caused by the retrovirus human immunodeficiency virus. It is it is a disease. It is a disease and caused by the retrovirus human immunodeficiency syndrome, immunodeficiency virus HIV, and characterized by profound immunosuppression that leads to opportunistic infections and secondary neoplasm and neurological manifestations. Opportunistic infection are infection that occurs more frequently and are more severe in people with weakened immune system, and induction peoples including people's HIV and opportunistic infection are considered acquired immunodeficiency syndrome defining conditions. Next one, the acquired immunodeficiency syndrome. AIDS was first recognized in 1981 in United States. The first recognition of AIDS in 1981 in United States. Secondary neoplasm. Secondary neoplasm, a malignant that arises from a pre-existing lower grade lesions or as a result of a primary lesions that has spread to secondary site or due to a complication of a cancer treatments. And neurological manifestations. Neurological manifestations include but are not limited to headache, impaired consciousness, stroke, seizures, meningitis, encephalitis, necrotizing encephalopathy, and Jaipur syndrome, and acute demyelinating encephalomyopathy. And this acquired immunodeficiency syndrome, AIDS was first recognized in 1981 in United States, and Centers for Disease Control and Prevention. Centers for Disease Control and Prevention reports the unexplained occurrence of pneumocystis jirovecii pneumonia in 5 previously healthy homosexual men in Los Angeles, or a Kaposi sarcoma with or without P. Zerovecii pneumonia in 26 previously healthy homosexual men in New York and Los Angeles. Homosexual means a sexual attracted to people of one own sex, and Kaposi sarcoma is a cancer that cause lesions in the soft tissues. And next one, etiology of AIDS. AIDS is caused by the HIV AIDS is caused by the HIV, a human retrovirus. HIV is a human retrovirus, and two types of HIV first and HIV second, which are genetically different but has related forms. The HIV first and HIV second are genetically different but related forms, and HIV first is most common type associated with AIDS in US. HIV first is most common type associated with AIDS in United States, Europe, and Central Africa. Next one, HIV second causes similar disease in West Africa and America. West Africa and India, and HIV second is transmitted less efficiently than HIV first. HIV second is transmitted less efficiently than HIV first. Next one, a structure. HIV is a HIV is a typical retrovirus. HIV is a typical retrovirus with a small RNA genomes of small RNA genomes of 9300 base pairs. Genomes means like DNA, RNA, gene carry, genetic informations. RNA virus have genome composed of RNA that encodes a number of proteins and is a spherical and contains nucleocapsid or surrounded by a lipid bilayer or envelope derived from host cell membrane. Nucleocapsid is the capsid of virus with the enclosed nucleic acid, and GAG encodes a polyprotein that is processed to release major structural proteins of virus. GAG encodes the major structural polyproteins GAG and is all the necessary and sufficient for the assembly non-infectious and immature viral-like particles. Paul encodes three important enzymes are RNA-dependent DNA polymerase or reverse transcriptase with RNase H protease in integrase. Paul gene encode are critical for the synthesis and integration of wild DNA into the host genome and generation of capsid proteins. Next one, INAV encodes for large transmembrane envelope protein responsible for cell binding and entry. NAV encode. NAV gene encode is a viral gene that encode the protein forming the viral encodes, and several small several small genes encodes several small genes encodes regulatory proteins that enhance viron production or combat host defense. Several small genes encodes regulatory proteins that enhance viron productions or combat host defense. This is the anatomy of acquired immunodeficiency syndrome virus. That AIDS virus contain the RNA and glycoprotein 41 envelope proteins and glycoprotein 120 envelope proteins, and this is the P17 matrix proteins, and this is the lipid membrane, and this is the P24 capsule proteins, and this is the reverse transcriptase. Next one, pathogenesis of AIDS. Pathogenesis of AIDS. HIV can infect many tissues. There are two major targets. HIV can infect many tissues. There are two major targets. First one, immune system, and immune system, and second one, central nervous system. These are the two major targets. First one, immune system, and second one, central nervous system. The primarily affected cell-mediated immunity. Primarily affect cell-mediated immunity. Cell-mediated immunity is that arm of the immune response that does not involve antibodies but rather incorporate the activation of macrophages and natural killer cells, enabling them to destroy intracellular pathogens. And next one, primary results in severe loss of cluster of differentiation for cells and T helper cells and impairment of the functions of helper T cells. And next one, macrophages in dendrites. Macrophages and dendrite cells are also infected. Macrophages and dendritic cells also infected. Next one, life cycle of HIV. Life cycle of HIV steps involved. Steps involved in life cycle of HIV. First one step is invasion. First one step is invasion. Second one step is maturation. Third one step is release. First one step is invasion. Invasion is a movement of cell from their normal location to the uh, surrounding non-diseased tissues. And next one, subtype of invasion, attachment and fusion. An extra part or extension that is may be attached to something then perform and particular functions. Next one, fusion. Fusion, the joining of two bonds into a single unit. Next one, maturation. Maturation, the process of become mature, and reverse transcriptase integration. Reverse transcriptase integration and release. Reverse transcriptase, also called RNA-directed DNA polymerase and enzyme, encodes the genetic material of retrovirus that catalyze or transcription of retrovirus into DNA. Next one, integration. Integration, the action or process of integrating and budding become larger a part of normal growth. Exit form cell. Exit form cell is when cell stop dividing and enter a quiescent state. Common sign and symptoms of common sign and symptoms of AIDS, severe impairment or suppression of immune system, severe impairment or suppression of immune system, pneumocytosis, pneumonia is most commonly seen. PCP, PCP, pneumocystis carni pneumonia is a serious infection caused by the fungus pneumocystis xerovici and of opportunistic infection. Opportunistic infection are infection that occurs more frequently and are more severe in people with weakened immune system. And CD4 cells, T cells count falls below 200 cells per micrometer cube of blood, and in healthy adults its value is 600 to 1500 cells micrometer per cube of blood. Next one, pharyngocytes, fat loss, pharyngocytes pain is a pain or irritation in the throat that can over with without swelling, often accompanies infections such as cold or flu. And neurological symptoms like partial or complete paralysis, muscle weakness, partial and complete loss of sensation, severe and difficulty in reading and writing, and poor cognitive ability, and unexplained plan and decreased alertness, rashes, headache, fever, lymphadenopathy. That are the signs and symptoms of AIDS. Next one, mode of HIV/AIDS transmission. Mode and mode of HIV/AIDS transmission like sexual contacts via the placenta and multiple uses, syringe and blood transfusion. That are the modes of HIV/AIDS transmission. First of all, through body fluid, through bodily fluid. Both bodily fluid cause HIV/AIDS, like by the semen, vaginal fluid, breast milk. That are the bodily fluids that cause the HIV. And HIV enter the bloodstream through open cuts, cracks in the skin, mucous membrane, and direct injections. That are the entries the bloodstream of HIV virus. Next one, tearing needles without sterilizations and through sex like intercourse, oral sex, anal sex are cause the HIV/AIDS. And mother to baby during pregnancy, transplacentally transmitted. During pregnancy, transplacentally transmitted, and drug postpartum period. During postpartum period, through contamination with maternal blood and infected amniotic fluid or breast milk. Amniotic fluid or amniotic fluid. Amniotic fluid is a clear, slightly yellowish liquid that surrounds the unborn baby fetus during pregnancy. Next one, diagnostic tests of AIDS. Diagnostic test of AIDS. First of all, blood detection test. Blood detection test, enzyme linked immunosorbent assay. ELISA. ELISA is enzyme ELISA enzyme linked immunosorbent assay and enzyme immunoassay is a that detect and measure antibodies. ELISA is a that detect and measure antibodies in your blood, and this test can be used to determine if you have antibodies related to certain infectious conditions. And next one, radio immunoprecipitation assay and indirect fluorescent antibody assay. RIF-IFA is a lysis buffer used to lyse cells and tissues for the radio immunoprecipitations. And next one, polymerase chain reaction. PCR. Polymerase chain reaction is a method and widely used to rapid make millions to billions of copies of a specific DNA sample and allowing scientists to take a very small of DNA and amplify it to a large enough amount to study in details. And last next one, Western blot confirmatory test. Western blot is the confirmatory test of AIDS, and Western blot test is used to confirmation or positive HIV diagnosis. Next one is urine test. First of all, urine Western blot test, and as sensitive as testing blood and safe way to screen for HIV and can cause false positive in certain peoples at high risk for HIV. Next one, Horatius. Horatius, the only Food and Drug Administration approved HIV antibodies as and as accurate a blood testing and involve collecting secretions between the cheek and gum and evaluating them for HIV antibodies. Next one, treatment of HIV. Treatment of HIV. First one category is nucleoside and nucleotide analogs. Nucleoside and nucleotide analogs like example abacavir, didenosines, emtricitabine and lamivudine, stavudine and tenofovir and gelcitabine. That are the example of nucleoside and nucleotide analog drugs. Next one category is non-nucleoside reverse transcriptase inhibitors, NNRT. Non-nucleoside reverse transcriptase inhibitor examples like dilaviridine and efavirenase and itravirine, nevirapine. That are the examples of non-nucleoside reverse transcriptase inhibitors. And next one category, protease inhibitors. Protease inhibitors like umprenavir and atazanavir and duronavir and fosmaperenavir, indinavir, lapinavir, ritonavir and nalfinavir, ritonavir and quinavir. That are the examples of protease inhibitors. Next one, fusion inhibitors. Fusion inhibitors like example infuviritide. Infuviritide is the example of fusion inhibitors. And next one, chemokine karyoreceptors antagonist like example miravirog. And last one category is integrase inhibitors. Integrase inhibitors are the example is reltegravir. Valtegravir. Integrase inhibitor is the drug is Valtegravir. Next one. Recent therapy for AIDS. Recent therapy for AIDS like drug class entry inhibitors, CCR5. CCR5 means C chemokine receptor type 5 and entry inhibitors, integrase inhibitors, maturation inhibitors, and non-nucleotide reverse transcriptase inhibitors and nucleotide reverse transcriptase inhibitors. And recently approved entry inhibitors like Meraviro, August 2017, and recently approved integrase inhibitors, Reltegravir, October 2017, and recently approved non-nucleotide reverse transcriptase inhibitors, example, Itravirine, Zen 2008. And next one, phase third is entry inhibitors like Vicryvirog, and phase second category is integrase inhibitors like Alvitagravir, and next one category phase third is non-nucleotide reverse transcriptase inhibitor. Phase third drug is rilpivirine, and phase second entry inhibitor example is Pro 140, and Pro 140 is a humanized form of RA14 antibody. And next one, TNX 355. TNX 355 is a safety pharmacokinetics and antiretroviral activity of multiple dose of ibaralizumab. And next one, maturation inhibitor is phase second drug is Deviramate. And last one, non-nucleotide reverse transcriptase inhibitor phase second drug is KP1461 and Resivir, and last one drug is Alvucitabine. That are the recently therapy for the AIDS. Next one, gene therapy. What is gene therapy? Gene therapy is a technique. Gene therapy is a technique that modifies a person's genes to treat or cure disease. And RNA interference. RNA interference is damage to a certain RNA sequence with participating of a different binding RNA molecules, and this system prevent viral infections unless virus had learned to cut it off in the course of evaluations. And researchers from countries including like Russia are developing two artificial RNA interference systems, and it's it is non-injurious to the patients and due to high specificity of action and does not damage its own RNA in cell infected by the virus. Next one, the fight HIV, Russia biologists have created three genetic structures. The to fight HIV, Russian biologists have created three genetic structures. Next one, these structures contain short nucleotide against sequence that find the most conservative molecules amongst all RNA molecules, that is sequence that do not change quickly and are important to the virus. And the sequence are the damaged in HIV during the gene therapy, and genetic structures significantly suppress viral reproductions. Next one, vaccine. Difficulties, vaccines of HIV, difficulties in developing and HIV vaccines. First of all, classic vaccine mimic natural immunity against reinfections generally seen in individual recovered from infections, and there are almost no recovered AIDS patients. Next one, most vaccines protect against disease, not against infections. HIV infections may remain latest for long periods before causing AIDS. And next one, most effective vaccines are whole killed or live attenuated organism and kill HIV. First dose not retain antigenicity, not retain antigenicity, and the use of live retrovirus vaccines raises safety issues. Antigenicity. Antigenicity is the ability to specifically combine with the final product the immune response. Next one, most most vaccines protect against most vaccines protect against infections from mucosal surface of the respiratory or gastrointestinal tract. The great majority of HIV infections is through the genital tract. The protect against the infection through the mucosal surface of the respiratory or gastrointestinal tract, and the great majority of HIV infection is through the genital tract. And first, in which first first, most initial approaches phase first, most initial approaches have focused on the HIV envelope protein. Most initial approaches have focused on the HIV envelope proteins, and 13 different glycoprotein 120 and glycoprotein 160 envelope candidates have been evaluated, and most researchers most researchers focused on glycoprotein 120 rather than glycoprotein 41 and glycoprotein 160. Next one, phase third, ADSWAX, ADSWAX vaccines, ADSWAX vaccines, and this is the first successful HIV vaccine trial in history. ADSWAX vaccine is the first successful HIV vaccine trial in history, and the percentage rate reduced to 26% compared with those who had been given a placebo. 26% are rate reduced 20%, 26% compared with those who had been given a placebo. Next one, planned clinical trials. Planned clinical trials. Novel approaches including modified vaccina, Ankara, MVA. MVA means modified vaccina Ankara, and adeno-associated virus. Adeno-associated virus and Venezuelan equine encephalopathy. VEE. VEE means Venezuelan and equine encephalitis. Next one, that are the references for the topic of AIDS. First one reference, the pharmacological basis of therapeutics by Gottman and Gilman's 11th edition. And next one reference is pathologic basis of disease by Robin and Colton 7th edition. And next one reference is pharmacology by Rag and Hell 6th edition. And last one reference is www.google.com. That are the references for used in the topic of ads. Next one, multiple choice questions of this topic. First one question, when was HIV first recognized in the United States? When was HIV first recognized in the US? Option A 1970, option B is 1975, option C is 1981, and option D 1986. The right answer is C. The 1981 HIV first recognized in the United States. Next one question. Which of these is a symptoms of HIV infection? Option A, swollen lymph nodes. Option B, fever. And option C, tiredness. And option D, all of the above. The right answer is D, all of the above. Swollen lymph nodes, fever, and tiredness. That are the symptoms of HIV infection. That are the infections of HIV, symptoms of HIV infection. Next one, a person a person has AIDS when which of these occurs? A person has AIDS when which of these occurs? Option A, exposure to HIV. Option B, HIV antibodies are found in the blood. Option C, the CD4 cells count is lower than 200, or opportunistic infections develop in an HIV infected person. And option D, a person has HIV for 5 years. The right answer is C. The CD4 cells count is lower than 200 on opportunistic infections develop in HIV infected persons during the AIDS. Next one question. What does HIV positive means? What does HIV positive means? Option A, either antibodies against HIV or the virus particles themselves are present in the blood. And option B, you have been tested for HIV. And option C, your white cell count is high. And option D, you have been informed about HIV. The right answer is A. HIV positive means is either antibodies against HIV or the virus particles themselves are present in the blood. Next one question. HIV attacks a certain kind of cell in the immune system, which it is. Option A, red blood cells. Option B, white blood cells and are called T cells. And option C, platelets. And option D, epithelial cell. The right answer is B, white blood cells called T cells. Right answer for the attacks on immune system. Next one question, which is the CD4 T cells count at which AIDS is considered to have developed? Option A, below 1000 per cubic milliliter. Option B, below 500 per cubic milliliter. And option C, below 200 per cubic milliliter. And option D, below 50 per cubic milliliter. The right answer is C. Below 200 per cubic milliliter is the right answer. During the AIDS, CD4 count cell is below 200 per cubic millimeter. Next one question. The risk for HIV/AIDS is tied to behaviors. Which of these behaviors can put you at risk? Option A, spending time with someone who have AIDS. Option B, not wearing latex condoms during sex. Option C, injecting drugs. And option D, both B and C. The right answer is D, both B and C. not wearing latex condom during sex, and injecting drugs are cause the HIV AIDS and risk factors for the HIV. Next one question. Why is a combination of medicine? Why is a combination of medicines called a cocktail used to treat HIV? Option A, the virus change mutates rapidly. Option B, each person responds to each medicine differently. And option C, combining medicines triple their strength. And option D, A and B. The right answer is both D, both A and B. The virus change mutates rapidly and each person responds to each medicine differently. The combination of medicine are called cocktail. Next one. What is the best way to protect yourself against HIV? Option A, get yourself vaccinated for HIV. Option B, use birth control pills. And option C, use a latex condom during sexual intercourse. Option D, both B and C. The right answer is C. Use a latex condom during sexual contact are the best way to protect yourself against HIV. Next one question. A person has had switch when which of the occurs? Option A, exposure to HIV. Option B, HIV antibodies are found in the blood. Option C, the CD4 cell count is lower than 200 or aplastic infections develop in a HIV infected persons. And option D, a person has HIV for 5 years. The right answer is C. The CD4 cell count is lower than 200 or aplastic infections develop in HIV infection person. Next. Thank you. Thank you all of you for a wonderful session.